All right. So today's lecture kind of changed a little bit as I was putting it together and reviewing what you had learned throughout the course so far. I was going to go through kind of go through Earth's history and show you at different times how microorganisms had impacted the Earth system and climate, ecology, and so on. But as I started looking at this global climate change example and how microbes have adapted to that and how they will help shape it, um, it was pretty easy to dedicate this entire class just to that particular topic. Um, and the cover slide shows how bacteria actually replicate. Does anyone in the class know what that process is called? I know there's some biologists in the class. Bacterial replication. It's starts with a B, binary fission. So growth rates are very important when we start talking about the different ways that climate change can impact microorganisms. And we'll show a few different ways in which it can impact the growth rates. So we'll start by a review of climate change as a coastal and global issue, and then we'll progress into understanding how microorganisms in coastal environments will be impacted by climate change, then go into the more open ocean idea, which is not far from here. It's not far from us here in the Gulf of Mexico and Louisiana. Um, so the major effects. Um, so climate change is going to shift our weather patterns. The two figures on the left, the A and B on the left, are giving a probability of occurrence of a weather pattern. And I'm missing the mouse, so I can't point at it. I'm sorry, I don't know why that's missing. So A, we have the temperature, and you can see the bell curve. That's the distribution of temperatures. You can see there's a natural distribution of normal temperatures with a few hot temperature extremes and a few cold temperature extremes. Now, the idea behind climate change is that this entire bell curve will shift to the right here. You see the increase in probability of extremes in weather climate, where you're going to end up with more of the hot weather and you'll have more of the record hot weather because you'll shift that entire probability of occurrence to the right. So precipitation, it's not a bell curve per se because of how precipitation works. A lot of the rainfall we get is light, so the probability of light rainfall is high, and then you have heavy precipitation at the extremes, and you can see that highlighted in the green on the right side of the figure. So you see very little of that extremes. Now, when we've shifted our climate and we've shifted that temperature, it's going to result in more precipitation. Look at the new climate curve. It leaves for much more heavy precipitation. Temperature increases evaporation. Evaporation should increase rainfall. Not in all places, but in many places it should increase the total amount of rainfall. So then you can also start thinking about the broader scale implications where temperature melts ice. There have been several examples of Arctic ice and Antarctic ice melting and big chunks of it dropping away. This can help cause the sea level to rise. And the precipitation can also end up causing more flooding, as we know all too well here in Louisiana. So those are some of the major effects and how they might be changing the global climate. But when we look in coastal regions, this is a extrapolation made by the National Oceanic and Atmospheric Administration. It's kind of scary. It shows a 6 feet rise in sea level by 2100 and its effect on Louisiana. And you can see the Gulf of Mexico is just really encroaching on the land, and you know the Gulf would actually go up to meet the West Baton Rouge Parish in this case. You can see that Lake Pontchartrain would double in size, swallowing out Lake Maurepas. Most of New Orleans would be protected only by the levee system in that case. And so as you get this much land swallowed up, it's going to influence the carbon cycle. It's going to influence the microorganisms that are living in that soil. And I really want to devote some time now into exploring how it's going to influence these microbial processes. Can anyone think of any microbial consequences, particularly in the coastal environments, of this much water coming in? Any ideas?
They might multiply.
They might, yeah, they might multiply. They're gonna get, it's gonna change the condition entirely. So you might have the multiplication of different microbial groups that do different things and result in different carbon releases. And that's where we're really going to focus this work. But we'll go through in turn some of the consequences for microorganisms. I borrowed this particular figure from Singh in a Nature Reviews Climate Change article. He had written it for the terrestrial environment, but I just changed a few words, and it's entirely applicable for what we might experience in a coastal environment. There are biological variables which can influence climate change. You can see the connected circles where I've changed phytoplankton and zooplankton community because they're critical for primary productivity, and that's how the carbon gets drawn into the ecosystem. But there's also microbial communities that live in the ocean as well that interact with the phytoplankton and zooplankton communities. Not shown in this figure is also grazers, which greatly depend on both of them. And then all of this actually carbon that gets introduced into the ecosystem eventually gets sedimented out into the sediment, and you have a sediment geochemistry signal where you support a microbial community in the sediments as well, which we've talked about a lot in my lectures. So the microbial adaptation and feedback response is going to drive an ecosystem response, which can include a greenhouse gas flux. You guys actually talked about that with Dr. Roberts a bit I guess it was the last lecture on Tuesday. He talked a lot about two different greenhouse gas fluxes. Does anyone remember those critical gases that he talked about? One of them is right on the slide. It's talking about CO2, which you've heard a lot about. The other one is very important. It's methane, right? So you have this ecosystem response, it can be related to, instead of impact of ultraprecipitation, it can be related to the impact of a change in carbon inputs to sediments. It can be an impact of sea level rise, where you are now soaking the sediment that's never been wet with seawater or fresh water before, and it's activating a different community, as someone had just brought up in the previous slide. There's an impact of temperature as well. The CO2 impact is related to ocean acidification. If you produce more CO2, you can have ocean acidification, which I think someone is going to talk about at some point in this course as well. So let's get into detail about a couple of these particular consequences that could exist. One is temperature. I say temperature of the land here. I mean temperature of the coastal region, whether it's freshly inundated soil or not, it doesn't matter. The temperature is very important. If you increase the temperature, you have an impact on microbial processes. Different microbes have different temperature optimums. When we culture microorganisms in the lab, we typically look for an optimal temperature at which to grow them. And you have probably heard the term thermophile, which means you can live at a higher temperature, or mesophile, which is you like a middle temperature, and we'll talk about that a bit later as well. Also, within these microbial communities, as you increase temperature, you increase the microbial activity, the metabolic activity. That means if you're producing CO2 as you respire oxygen, you're going to do that at a much faster rate as you increase the temperature. And that was introduced to you early on in the course as the Arrhenius equation. And it's very valid for microorganisms as well. They're going to produce more products with more temperature until they reach their temperature optimum. So, sorry for any background noise. There's apparently work going on outside of my window too. You have saltwater intrusion into land, and why does that matter? So just like there's a temperature optimum for many microorganisms, there's also a salt optimum. Some can stand more salt than others. By salt, I'm talking about seawater salt like NaCl. It's critical to have your osmotic pressure right in the cell. Some can regulate that better than others. Electron acceptors. So as you dump salt water or ocean water into fresh water or into the land, which is going to happen in coastal regions as the sea level rises, you're going to increase the abundance of a different group of electron acceptors. You're going to have sulfate added in a huge abundance, and some nitrate as well, which these weren't really there so much before. They were in trace amounts, but as we've discussed a lot in the course, seawater is just loaded with sulfate. Then you also have the instance where you might have forests that are now covered with water, and they had their own carbon, their own soil communities there. You have all the leaf litter, the trees. That's all actually carbon that can serve as electron donors for microbial processes that were less accessible to microorganisms when the land was not soaked. So these are a couple of concrete examples, and we're now going to go into depth about them a bit more. So we talked about temperature in microorganisms. So this plot shows the growth rate of bacteria, and you actually have the temperature on the x-axis, and it goes about -5 degrees to 20 degrees for psychrophiles, and you'll see the shape of the curve for psychrophiles. So that means when you're at -5 degrees Celsius, the growth rate is going to be really slow for a psychrophile, almost not existing. And that means that their metabolic rate will be very low as well. And then they will peak around 10 degrees Celsius. So that would be like their maximum growth rate. And then again, once it hits 20, they're going to go down again because it's just simply too warm. They're not adapted to that particular temperature. So if you think back to the Arrhenius equation, between -5 and 10 degrees, they ramp their activity up tremendously. And then they just get too warm, and that function actually doesn't apply for that particular population. Next, you have what are called mesophiles, or mesophiles, however you want to call it. They're functioning from 15 to about 45 degrees Celsius. And again, you see the sharp ramp up in activity. You see higher activity in comparison to psychrophiles. And thermophiles, again, you see quite higher activity. The thing is, at higher temperatures, microorganisms have to replace their biomolecules at a faster rate because biomolecules are simply less stable at higher temperatures. The upper end for life or microorganisms shown so far is around 120, 121 degrees Celsius, which is kind of ironic considering we autoclave everything at 120 degrees to sterilize things. So taking temperature a step further, we can actually quantify the response of particular microorganisms in what we call a Q10. This is for each 10 degrees Celsius, you have a certain response in the microbial activity. And this is interesting because it can give organism specific response. So in seawater, we went through this plot before. Ooh, don't know why that happened. Sorry. You have the sediment. You've seen this plot before. It's overlying seawater, and you have the cascading series of electron acceptors. Here I call them terminal electron acceptors because that's an important concept that I'm going to kind of introduce in the context of climate change, where you have oxygen progressing through to nitrate, iron, sulfate, CO2. So the rates of these processes, whereas you have like sulfate reduction, you would look for the production of CO2 from sulfate reduction. If you have CO2 reduction, CO2 is your terminal electron acceptor, you actually start producing methane. So your rate would produce methane. So if we look at our ratio of rates, that's looking at rate 2 over rate 1 at two different temperatures, T2 minus T1. And you can see that there's a lot, like an exponential increase in rates with respect to temperature differences. And that's an important concept to grasp, and that's the Q10. So very important for our purposes is the temperature influence and how you produce CO2 versus producing methane. The figure on the right, again, is borrowed from the terrestrial environment, but actually the same thing applies in marine sediments or coastal sediments as well. You can see these polymers and monomers. So this would be your high molecular weight organic matter and your low molecular weight organic matter. They have to be hydrolyzed or fermented to produce hydrogen acetate or lactate, or I think are a series of fulvic acids, which is the SCFA. So you have to get these smaller moieties to actually support other microbial processes. So that's like the initial step that supports the entire microbial community in sediments to a degree, because these polymers and monomers are actually produced by primary productivity. And you'll see two sides here. You'll see a minus sulfate, which means no sulfate, and you'll see a plus sulfate on the left-hand side, which is with sulfate. So when you don't have sulfate, you see the arrow shifting toward methanogenic archaea. And as you remember from the previous lectures, methanogenesis is one of the few processes where only archaea actually have been shown to produce methane. And this methanogenic archaea are responsible for 30% of the global methane emission. So it's interesting that it shows no sulfate there. But then with sulfate, you have sulfate-producing microorganisms, and you see the production of sulfide, and also it would produce CO2. And you'll remember the figure from the left from our last lecture, where we looked at production methane by methanogenic archaea, it says bacteria, because we used to actually think it was bacteria. Anyways, so for that methane to bubble out of seawater, it actually has to escape where the sulfate reducing microbes are. So why should sulfate reduction even matter for methanogenesis? Why is this all lumped together? Um, these are really two terminal electron accepting processes that are drastically different. One relies on CO2 reduction, the methane, the other relies on sulfate reduction, and it produces CO2. So why does it matter? So this is a really classic paper. Clearly it's from Lovely, 1982, but it's so classic and it points out something so very almost universal in marine sediments that it's still very valid today when you think about methanogenesis and sulfate reduction. So he did these experiments where he looked at the hydrogen concentration over a period of time. You see time on the x-axis, hydrogen pressure on the y-axis on the left, and you see micromoles methane produced on the right. And he noticed that when you had conditions where sulfate reduction was not favorable, so what he actually did was add, you'll see this MoO4, that's called molybdate. Molybdate actually, it confuses sulfate reducers. They think that it's sulfate, they grab it up, but they can't actually make energy from it, so there's no sulfate reduction. And when you look at sulfate reducers and you have the molybdate there, that actually allows the methanogen to be active. And you'll see the hydrogen partial pressure hangs out around 10 to or 8 to 12. He has it here before it really comes to a point. If you look at conditions that are favorable for sulfate reduction, which would be the dashed lines, and you can look at the circle dashed lines, and you'll see that they really just suck the hydrogen down. This sets up a situation where you have two different microbial communities competing for the same substrate, and one outcompetes the other. Who do you think outcompetes who for hydrogen? Any ideas, class? If sulfate reducers push the concentration of hydrogen down very far, who can outcompete who? So that means the sulfate reducer is the winner, right? So the process, if you wrote it out for methanogenesis with CO2 reduction, it's often called hydrogenotrophic methanogenesis. Is CO2 plus hydrogen goes to methane plus water. For sulfate reduction, if you're looking at sulfate plus hydrogen, goes to sulfide plus water plus hydrogen. So,
Marshall, there's a question. The question is why, I'm sorry, is that why anaerobic breakdown of contaminants in the soil is so slow?
In terms of competition, no, I don't think that would be why. A lot of contaminants are broken down not from methanogens because they're handling only CO2 and some very small carbon-containing molecules. The rate-limiting step might be the fermenters that actually break it into the compounds that are used by sulfate reducers. So there's one step above that probably has to be utilized before it gets to a sulfate reducer or a methanogen actually using it. And actually, if you have fermentation, you actually produce hydrogen as well. And sulfate reduction with hydrogen is highly energetically favorable. A sulfate reducer gets a lot of bang for its buck from that. So it would be the step ahead of that should probably limit the remediation process. So the hydrogen threshold for sulfate reducing microbes is about 2 nanomolar. It's not from this publication, but I'm just giving it to you, versus 10 nanomolar for methanogens. So you have what's called substrate level competition, and they're competing for the hydrogen. And that really shapes what we know about marine and coastal ecosystems with respect to methane releases as well. So not only do you have this situation where methane can only be produced when no sulfate is around, but you also have a situation where the methane to get released actually has to escape the sulfate reduction zone. So now I'm throwing another curveball at you guys, and this is where things get really interesting. As you go through marine sediments, where you have a sulfate reducing zone, you have a specific process that occurs typically at the base of the sulfate reducing zone. So where this sulfate is, I wish my arrow worked here. I don't know why it doesn't. So once the sulfate gets starting to be reduced, and where you have the methane that's diffusing into the sulfate reducing zone from this anoxic methanogenic sediment, you actually have sulfate used as an electron acceptor to oxidize methane. This is a particular process that confused scientists for, I guess, 30 years now. It's been a very hot topic because it makes little sense. The energy yield from this reaction is very low, and the idea was that you had an archaea that was performing methanogenesis in reverse and was sharing an electron acceptor or electrons directly with a sulfate reducer. So you had a case of symbiosis, you could think of it, so microbial symbiosis. And if you split the energy yield between two microorganisms, it's really not even favorable. We talked a lot in the first class about what makes a reaction possible, and to pump a proton across the membrane, you're looking at about negative 20 kilojoules per mole, and you would have to get negative 40 because you potentially have two different organisms that should profit from this reaction. And most of the time, in most environments, the total reaction would yield less than, or actually greater than, negative 30 kilojoules per mole, making it really not feasible. Now, advances have been made, and we have a better understanding of this process now. In some cases, it seems like it's only one organism that's doing it, but the critical thing for you guys to remember is that for methane to be released from marine sediments and coastal sediments, you have to escape that sulfate reduction in most cases. If the concentration is high enough of methane, then you can actually escape it. But those are rare cases, but they do happen in the coastal environment actually, due to the fact that there's so much carbon coming out into the coastal environments from the rivers like the Mississippi. You just get so much electron donors dumped into the sediments, you can actually exhaust your sulfate stock to bring up those electron donors before you get to the methane. So I've digressed a little bit, and let's get back to this temperature influence on CO2 versus methane production. And really, this is just reiterating what I just said. I wanted to solidify it for you guys. To produce the methane, you got to have no sulfate for the methane to escape into the atmosphere, which in coastal areas you would look for something like on the left where you have less and less sulfate has to be present to actually allow the methane to escape and exchange with the atmosphere. So this gives you an idea for how complex these systems are and how complex these problems are, and also how understanding how microbes as a community will respond to different situations is really important. And again, we can go back and talk about something that Dr. Roberts talked about on Tuesday. It's really important to think about this terminal electron acceptor for something like carbon dioxide and methane emissions, because methane emissions mean so much more in some ways than carbon dioxide does. 1 unit of methane traps as much as 23 units of carbon dioxide. And we've seen the concentrations of methane increasing in the atmosphere as well, particularly over the past 10 years, which are not even displayed on this particular graph. The concentrations of methane have just ballooned. And there's different theories for why that's happened, but coastal environments are something we really need to keep in mind when we consider this, and how that water and that freshly inundated soil or sediment will actually respond to these So as I had mentioned before, one way that we look at this stuff is looking at a Q10. We look at we think about the Arrhenius equation, how particular microorganisms respond to increases in temperature. So there's been a lot of studies on this particular aspect. This one is from Fu et al., and this is from a wetland actually in Tibet, maybe. And he did some interesting studies on the sediments alone to see how temperature changed the relative release of methane or CO2. So he's looking at these very processes in sediment that we're talking about, but in a wetland setting. And he's subjecting the sediments to a variety of temperatures. He's plotted it -1 over RT, which R is just the Boltzmann constant. So if you go more positive, you're actually increasing temperature there. And he has the LMP on the y-axis, which is the natural logarithm of the product, and the top graph A, that's methane. So you can see that methane responds from -84 to about -77. You can see that CO2, on the other hand, only goes from -79 to about -77, which means that methane releases go up faster than CO2 releases as you increase the temperature. I don't think there was any sulfate in that wetland in particular, or at least there would be very little. So there's not much competition for methanogens in that case. In the ocean, we would have to consider sulfate reduction. The Q10 for methanogenesis, I looked at one paper and grabbed some ranges, was ranging from 2.5 to 6. For sulfate reduction, I saw a Q10 of about 2.5. But it could go higher, of course. So if you're responding about like 6 orders of magnitude, that's your response rate to temperature increases, you're probably going to blow sulfate reduction out of the water, which means that we should really think about how these coastal ecosystems will change as we increase the temperature, and also wet them with seawater and activate more carbon. I showed the Arrhenius equation here. You don't have to memorize it. You don't have. I'm not really going to question you on that particular aspect, but I wanted you to be aware of it and what it looked like. The important thing is that it gives a exponential response of microbial communities with respect to temperature and the product they produce, whether it be methane or CO2. So again, what I also mentioned, when the ocean is intruding into our coasts, which are somewhat brackish and sometimes fresh, depending on how far inland we go, we actually introduce a lot of electron acceptors. This is salts and seawater. Chlorine and sodium, they're not fueling microbial activity directly, but that sulfate is very important. You can see it's 7.7%, and that's by weight, and it's approximately 28 millimolar in seawater. To give you some perspective, sodium and chloride are about 555 millimolar. Oxygen is, if it's saturated at a lower temperature, you're looking at about maybe 300 micromolar. Nitrate, I also mentioned earlier, that'll hang around 30 micromolar, but there are special situations and instances where that can reach as much as a millimolar. So we're basically feeding that sulfate into the land that might not have had that sulfate before. And maybe that does shape our methane emissions. Again, this is getting at my very point here that the ecosystems are complex and you can't just look at one variable alone. So now I'm going to go through a couple studies that have been done in different sediment environments, and then we'll jump to the ocean after that. This is by a study done by Nat Weston, where he actually used some really interesting techniques called a benthic plug flow reactor. I can talk to anyone after the class about these particular methods if they desire to stick around. So he was looking at terminal electron acceptors in salt marsh sediments from the coast of Georgia. And you'll see this much more elaborate flow chart where you have the primary producers that make the particulate organic matter. And this is exactly what we were talking about before in the figure from the terrestrial environment we showed. The particulate organic matter must undergo hydrolysis to become high molecular weight or dissolved organic matter. And then you have to have hydrolysis or fermentation, which is the stuff I alluded to earlier as being a critical process for some remediation steps, bioremediation processes. Anyways, the dissolved organic matter will get switched over to a labeled dissolved organic matter, hopefully. And then you have what's called a terminal metabolism, which could be, as we discussed earlier, CO2 as a terminal electron acceptor, or you could have sulfate reduction, and that would shape whether you release CO2 or methane. What escaped the system entirely was buried, become a long-term burial carbon. Incubation. What I want you to take home here is that he did these incubations at various temperatures from 5 to 35 degrees Celsius. I guess about 33 degrees Celsius. And there's a lot of points here and a lot of data, but one of the steps he was really interested in looking at was how this label dissolved organic matter was produced, because that's going to be what fuels either your CO2 or methane production. And what he noticed was that before even considering how much methane or CO2 are produced, if you just looked at these these small carbon-containing molecules, volatile fatty acids for the chemists in the group, he saw this dive in the production of volatile fatty acids, which means that you stop actually feeding processes like sulfate reduction and methanogenesis at these higher temperatures. So you actually formed a roadblock and carbon degradation before you could actually get the terminal electron acceptor. And I think that's a very interesting observation. And again, it points out the complexity of these ecosystems.
Yes, and we'll be open tomorrow as well. We're just closed on Monday and Tuesday. My pleasure. Yes, you come by and see us.
Can somebody turn off their mic? Somebody's got a hot mic.
You have a great day.
From the ocean down to the sediments now, we'll look at the kind of influence of the ocean on marine sediments. So if we look at the figure on the left, we have a current and future situation. And this is kind of accounting for warming and also acidification of the ocean. You can see the CO2 exchange is bigger, and you can see this drop in pH from 8.2 to 7.8. You can see the warming from 15 to 20 degrees Celsius in surface water. And you can see this mixed layer depth actually goes up higher in the future, which means you're doing less to get nutrients up to the surface because the stratification becomes more intense in those cases. And that affects your primary productivity. That affects what can actually get into the deeper waters to support microbial process and marine sediments. The figure on the right actually shows how you influence the system as well, and you actually decrease the intensity of the carbon cycle in the ocean altogether. You can see the much bolder arrows that give the exchange of CO2 to DIC, and then the DIC buffer into calcification, which is forming your carbonate shells in some cases, and your phytoplankton as well. You can see there's greater organic carbon sinking in the current versus the future where the line's much thinner. So this is a feedback of the entire system, which slow microbial process in marine sediments based on slower activity or carbon cycling in the surface ocean. So again, not everything happens in the black box. From the ocean down to the sediments, you have an influence of the nitrogen cycle as well. And you see its intensity increases very much, indicated by the size of the arrows. These are a number of processes that I know Dr. Roberts went over a while ago when the class was started. You can see the sequential denitrification steps are very heavy. But what happens is interesting is that the nitrification loop becomes much smaller. So you're replenishing the nitrate and nitrite at much lower levels than the other processes. And that alone can also greatly influence microbial communities that are thriving in sediments. So the microorganisms with a really high affinity for ammonium, which there is a particular group that do, there are archaeal nitrifiers, would actually be very much better suited for this situation versus the current situation. So you can imagine a situation where you shift from archaeal processes versus bacterial based on the influence of the nitrogen cycle in the future ocean. This was a real study by Sarmento where he was looking at the influence of warming on grazing in the surface ocean. And again, this is avoiding putting everything in a black box. He's including this grazing stuff. So you see the natural log of bacterial production on the y-axis, and then on the x-axis you have temperature. So as the temperature increases here, because it's not directly temperature, it's 1 over the Boltzmann constant time temperature. So as you have an increase in the temperature or the Boltzmann, you're actually decreasing the temperature. So you had an increased amount of bacterial production at higher temperatures, but you also increased the grazing, which is shown in B, which is the natural log of grazing on bacteria. So you had an intensification of the processes so far based on this one study and grazing of bacteria. Other people are trying to model how cyanobacteria will change locally once you increase precipitation and increase the temperature of surface waters. Two of the big ones are big organisms responsible for these processes are Prochlorococcus and Synechococcus. The left figure shows their abundance in present day in seawater, and not only their abundance, but their distribution. And then the right figure shows their relative change, their changes in cell abundance as a percentage. And what's interesting here is that the Prochlorococcus appear to actually disperse a little bit more. They get in a wider range across the ocean, become more uniform. The Synococcus actually contracts its distribution into more narrow bands across the mid latitudes. And these studies are based on what we know about how they function physiologically. So again, this is considering how they respond to nutrients, how they respond to temperature, how they respond to CO2. So these full-scale biogeochemical studies actually allow us to elucidate these future trends. And that is, so there's a big take-home message today. It's clear that the climate is changing, but as I keep on harping on, it's really impossible to look at individual variables alone because we are talking about complex ecosystems, complex microbial processes. We have to consider the entire system to really understand what the final impact of environmental change will be. So pure culture and single process analysis can be informative. We looked at the example of methanogenesis and sulfate reduction in marine sediments, and we saw how we could kind of uniformly understand which process would be occurring based on this hydrogen threshold because it's competitive substrate. If we could do more of that and understand different aspects of different communities to get a better idea of what happens in an ecosystem, it would be very helpful. But we always have to consider the whole picture. And that's really all I have for you guys today, and I will hang around for questions as they come up. Does anybody have anything?
Guess not, Marshall.
I guess it was boring then.
Or very good.